Welcome, everyone, to the N of One Collaborative Seminar Series. My name is Nicole Nolan. I'm N1C's Programs Manager. It's my pleasure to welcome you to today's exciting session. First and foremost, I want to thank our partner, the Chan Zuckerberg Initiative, for helping to support N1C's mission. Secondly, I want to invite you following today's talk to join us on Connections with any questions, follow-up points of discussion. We would love to continue the conversation with you there. I'll be dropping the link in the chat and hope that you'll register and come say hello. Importantly, I'm excited to share that you can also register for N1C's annual meeting on Connections. It's October 10th in Denver, Colorado, the day before OTS officially starts. So for those of you that are already going to be attending the Oligonucleotide Therapeutic Society annual meeting, consider extending your trip by one day on the front end. We would love to have you. With that, I'm happy to pass the mic to the moderator for today's session, Dr. Tobias Pullman, who's the lead of N1C's CMC working group. Tobias.
Yes, hello. Thanks, Nicole. Hello to everybody. So it's my pleasure to host the two speakers today. The first speaker will be Pablo Lores Lareo from Silentis. He's a master in chemistry and worked with oligonucleotides, nanotubes, and did a lot of analytics in his career. He received a PhD in chemistry, analytical chemistry from University in Berlin, and worked as a scientist in analytical chemistry and CMC at Silence Therapeutics before he was an independent CMC consultant. And now he's director of CMC at Silentis. So I'm happy to host you here. And our second speaker will be Seppe Hermans. He also received a master in chemistry from Leuven University and did analytical method development at Kila. And since 2 years, he's a PhD student at Vienna GMP and is working on a standardized method for small-scale GMP oligonucleotide synthesis. And he's volunteering in the N1C, organizing the CMC work group and organizing the N1C roadmap. So very welcome. Happy to have you here and looking forward to your presentations on the CMC roadmap and framework for N of 1 oligonucleotide therapeutics. Pablo, the mic is yours.
Thank you, Tobias. Thank you so much for the kind introduction. And welcome everybody to this session. Originally, this conversation started with Seppe during one of the conferences that we attended. As Tobias mentioned, I come from a background of developing therapeutics for big indications. And when talking to Seppe, we realized of how different it could be for like when we develop oligos for big indications compared to these kind of therapies. I'm gonna briefly share my screen. There we go. Are you seeing now my screen? Is everything fine?
Looks good, Pablo.
Perfect.
Okay, because I don't see you. Yeah. As Tobias mentioned, I started in biotech, then I was a consultant, and now I work at a CDMO. So that what gives me is the perspective of have seen lots of different molecules at different stages of development, and how they approach the development of an oligo. And my objective today with this talk is to prepare a little bit the framework of general oligos, so then each of you can go and apply it to your therapies. First, in regard to some of the words that we're always using and throwing back at you is CMC, it's chemistry, manufacturing, control. This is a department in biotech that basically manages how a molecule is done. It's quite descriptive by what it's said, but eventually the department has to provide all the information, physical, chemical, and biological characterization of the drug. And it covers the production, life cycle, and manufacturing, and all the control that we do. This is a department in all biotechs that is an enabling role. That means that clinical and non-clinical are driving the development of a drug, and we are just supplying the material by managing the external infrastructure that we have, the CDMOs, and then compiling the data to supply the documentation to regulators. So yeah, that's where we stand, and how we make sure that what we are providing to the material that we are providing to clinical trials is good enough. I just wanted to throw a bit more of a definition, which is GMP. I remember when Sepe came to me and he was like, he just said, most of the people who develop these kind of therapies, they are not used to the lingo, they are not used to, or if they have heard it, they don't really know what it means from a practical perspective. So I'm not gonna go deep into what GMP is. You would need like probably several weeks, and I'm not the most qualified person to do it. However, just as a brief introduction, so GMP is good manufacturing practice, and it's a framework that we have to show that we consistently supply something that is good quality, right? In a spirit, we have this definition here. If you can explain exactly what you did, prove you did it in the same way every time, and show that it worked as intended. If you haven't done that, you haven't really do it. Basically, this is the lab journal of your students, that how it should be done, but in a formal way. What it means, GMP, when applied to the real world, is a lot of documentation. It's documenting absolutely every single step you make. It's controlling every step you make. And once you have done that, and you've done your process, the fact that you're able to trace back every single decision, every single point, every single measurement, every single piece of data that you have to the source and the measurement, and eventually to reproduce that experiment. GMP, it's something that is thrown out as a broad term. And it's a framework, as I mentioned, that is governed by some general rules. However, each country has their own set of flavor or their language when talking about GMP. Probably you have heard of CGMP, which is how the FDA describes GMP manufacturing. You have heard GMP, just GMP, that is how we describe it everywhere else in the world. And as I mentioned, even if we have a set of common rules, then it's locally enforced by different administrations. So in the United States, the FDA. In Europe, we have the common framework by the European Union, but then each national organization is responsible from enforcing those rules. You also have in Japan, in China, their own associations. So the Pharmaceuticals and Medical Device Agency in Japan, or the Anvisa in Mexico, in Brazil, sorry. And so on. So each of the different regulators, they check their GMP. So when, as a CDMO that I am now, and I'm trying to produce drugs, oligos for different clients that I have around the world, I have to make sure that I am doing the rules that satisfy all those organizations. So not only a GMP framework, but also speak the different languages that each of them speak. So just to finish this brief introduction on GMP, the take-home message is that GMP is not about perfection. It's about consistency. So every single process is going to have mistakes or variations. And it's not that the GMP is going to make sure that they don't happen. It's that that definition that you have, you tolerate it. So you understand how your process works. Ergo, you can define where it was, how it happened, and what's the effect that it had. Good. That being said, let's move to my expertise, which is oligos. I mean, you have seen how they are synthesized. You understand that perfectly fine. We have a cycle of reactions. Each of them carries a set of variables, flow, concentrations, different identity of reagents, and so on. That's just for the synthesis, but then once you had it synthesized, you have to do the downstream, which involves activating the protection and several steps of purification until you have your isolated product. So I'm talking about GMP. I'm talking about having a process that you have well characterized and having a process that you are able to keep control. And then I add all this layer of complexity of millions of reactions that each of them can go wrong and millions of parameters that I have to control. So how do we apply this to oligos? It's the question. So in the case of a standard molecule, one that has to go through clinical trials, that one that has to address larger parts of the population, even if it's a rare disease, you have to go through clinical trials. And CMC, this department that is supplying the drug, has to always be one step ahead to the rest of the company. That means before you go into a phase 1, CMC has to be already manufacturing that material. When you go to phase 2, you need to have that material. So during phase 1, you have to be developing that. During phase 3, the same. And when you go to commercialization, you had to do that while you were developing. So it's an eternal fight of departments and egos, if I can say that, between clinical pushing to have material and supply it consistently, and then CMC trying to do that with all the constraints that we have. When we try to do this, when we try to address this, there are some maximas that we apply in each of the stage. So when we are in clinical development, what we do is we define the process under which is manufactured the molecule. Then during phase 1 of clinical trials, we start developing it, improving the process. During phase 2, we have a process that is developed. So then we characterize it and we understand how small variations affect the process. And finally, during phase 3, we do a process validation, which is another term that is being thrown out very broadly, but basically it makes sure that your characterization, your understanding of the process is correct. So yeah, let's go. I'm gonna drive you very, very fast and very, very shortly on how one of these programs is being done and where you should put your attention. So most of companies, when they arrive to me, they have a treatment. So a molecule whose biology has been validated. So it works as intended. It follows a proof of mechanism. It decreases a biomarker or it increases another biomarker. And then they have proof that the molecule is non-toxic in animals, and then they want to go and do some testing in humans. That's the most common package that arrives to me. So what I do for them is I do some impurity markers. I define the process as I told you, and some process development already to, I don't want to give them a process that is terrible, but a process that is somewhat already good. I develop some methods, the part of control that I mentioned before, transfer all that to the scalable platform for synthesis, and then do the manufacturing. Usually I do a GMP batch, which is a large scale and is the one that goes to clinical trials. And I do a non-GMP in the same scale. So same conditions as the GMP, but non-GMP. So if there are problems, I can correct it. And that is what eventually that's the work package that goes into getting a molecule to phase 1. So yeah, quite a standard. Good, then we give the material to the clinical development. They run their trial, and the trial is successful, and they want to go to a phase 2. How do we drive that development? And that's basically the part that takes the most of companies, because as I mentioned, this is a process that takes 1 million parameters, 1 million variables, a lot of different things. Where do you focus your attention on what to improve, how to determine that working space of your process? And well, as boring as we are, the process development is driven by a risk-based approach, so a risk assessment. We took the batch record of the molecule. This is a document where it says every single step, the conditions that you have, all the recipe of your oligo. And for each thing that can go wrong in each step, you write it down in a really huge Excel. That Excel, you then rated each of the parameters by severity, occurrence, and detection, give them a score, and depending on that score, you will get a criticality factor. And based on that criticality factor, so if you get something that is highly critical, then you need to do some process development. If it falls in the medium, you can recommend to do some process development, and if it's in the low, then you can leave it there. And what do I mean by this criticality? Well, as I mentioned, all parameters are being ranked based on severity, occurrence, and detection. If I have something that is easy to detect, it's going to get a low score, but if it's difficult to detect and happens very often and it has a strong severity, then I need to understand that process better. Or I can do things like improving the analytical methods. In that matter, then I'm going to decrease the uncertainty because I'm able to detect it better. Or if I understand under which conditions that has an effect, the severity is lower, then I can decrease the score. And that's the work that we are doing, as I mentioned. So process with a lot of variables, then for each variable, I write all the things that can go wrong. There can be a million experiments that I have to do, and this just allows me to set a tool to rank them and over time put more centrality on my process. And that's basically what we are doing with the results from that failure mode and effect analysis, the risk assessment that I told you, FMEA. I come to the batch record number 2 and the second line of specification. There are other points that I would like to mention here. Formally, the understanding of your process is translated into what we call the normal operating ranges, or NOR, and the proven acceptable ranges. That means under which conditions your reaction can happen and takes you to the okay, sorry, I was checking the message from Tobias. So your understanding of the process basically translate into those two things, the NOR and PAR, and eventually they translate into your process through the critical quality attributes, critical process attributes, and in process controls. So at the end of the day, understanding of the process needs to translate on understanding of the recipe and how you execute the recipe. Good. So that's how we drive the development. That happens from phase 1 to phase 2. Another thing that we should keep in mind when we are doing a phase 2 is that usually we are scaling up the process. This is not the same for your kind of therapies, but for normal oligos, you jump from instrumentation to instrument until you reach the final scale of your process. And a lot of different things go into that also. I mean, you have to risk not only the oligo part, but also the starting materials. If you're in the field, you have seen how discovery departments, they are getting more and more creative as a synonym of annoying, with more complex conjugations and different things that they want to put in the oligo. And all those supply chains need to be understood and characterized as well as we do the oligo. We need to validate the methods, so the methods need to be performing always under the same conditions. And then, yeah, once we have the final scale, we have the final methods, and we understand the process, we are ready to go to the validation, which is the phase 3, as I mentioned. This is probably the biggest piece of work for our CMC department. Validation is a term that is being thrown out very lightly around, and it's a very complex process that you have to do, and basically is covering all your understanding of the process. So you start with a validation protocol for your process. You throw all the conditions that you have studied. You show that they change as you said that they were going to change, and you compile all that data in a package that you give to regulators, and they go line by line and check like, okay, these people, they know what they are doing, or they don't know what they are doing. Yeah, I don't want to keep going much more because, as I mentioned, this is generalistic from an oligo, and is what eventually, as I mentioned, is the whole framework of all the work packages. I could go hours and hours and hours explaining each of them, but at least for you to have a general vision of what is done for a standard oligo, and then for sure the data package that you're going to have to show to the authorities for your therapies is not going to be the same, but it will have to be, you will have to defend it in a way that makes sense to them. And if you know where you're going, then maybe this allows you to then apply it to each of your molecules. Last thing that I want to show you is, yeah, as I mentioned, the methods at the beginning, they get developed and validated early on. The process development, you drive it early on and then define it in your process. And finally, one chapter that I completely ignored until now is stability. You have to prove to authorities that your molecule is stable, not only in short terms and in different formulations, but also what happens when when you freeze and thaw the sample, what happens when you put buffer on it. Those are data packages that a usual oligo would need to have. So that's something that you will have to evaluate with your processes, not just the powder per se, but under each formulation that you may have. I think with that, I'm good on time, Sepe. Not running longer than I was expecting. But yeah, just as a summary of my part, making an oligo is a complex process. To keep it under control, you need to have a great understanding of the process. How you drive that understanding and that development is through a risk-based approach. And eventually, that's the same that you will have to do when you're developing your molecules. The risk each molecule as much as possible to then convince the regulators that your therapy is safe for patients to use. That being said, Sepe, I give you the floor.
Maybe I would suggest Pablo, because I'm sure it's a big area and a big topic, especially for people who are not so involved yet in the GMP environment. Maybe we can discuss a few questions
Sure.
before Seba is taking over. Mattis, you had a question. Better you ask it directly because it's a bit complex. Maybe we can also separate the questions.
Yeah, thank you, Tobias. And Pablo, thanks for your talk. I just had a question. You suggested to take the non-GMP grade ASO for IND enabling studies and reserve the GMP grade ASOs for phase 1 onwards. That was written on one of your slides. My question is that really a good choice? Wouldn't you recommend to indeed already use the GMP grade ASO at least for the late preclinical IND enabling studies? For two reasons. I mean, you have the product you actually want to use in the humans, and rather than a different product. And secondly, it's the cleanest, at least most well-documented product. And that's the one you want to use in your late preclinical IND enabling studies.
Yeah, really good question. It's usually a matter of price. The GMP is going to be much more expensive than the non-GMP. However, there are other differences too. As I mentioned, the non-GMP, you have it index is the exact same scale that you are doing. And the benefits of doing that and using that first batch besides the GMP is that you can purify it in a way that is a bit more generous. So when you do the purification, you include more fractions in it. It's going to have more impurities, and you're going to do your tox with a product that has more impurities but is representative of your process than the one that you do with the GMP. Later on, when you're scaling up, impurities are going to change, and you want to make sure that you have included as many as possible in your tox studies. So it's a matter of not that much GMP, non-GMP. It's a matter of the impurities that give you freedom to operate later on. I shouldn't use freedom to operate, sorry. But having seen impurities in tox studies that will allow you to justify more batches.
Pablo, I would bring up another topic in this regard. That's stability of the product. Usually you want to use your GMP batch directly or as soon as possible for your clinical trial. And you have to have a bit of stability studies going ahead. And using the GMP batch for your last clinical or preclinical trials, it would, induce the risk that your GMP batch is running out of your stability data. So I would also advise to use a very good batch, a controlled batch, but not a GMP batch for the late preclinical trials.
Yeah.
Or Pablo, would you agree?
No, correct. I completely agree. And one thing that I said, also what you are saying is quite important, that well-characterized batch. The tox batch is probably the most important batch ever produced in the development of a drug. And you're going to gain understanding through development. Your methods are going to improve. You're going to be looking for other things. So it's very important to keep part of the material aside, just so you're able to characterize later on.
Mm-hmm. Eric, you're waving. You're still on mute?
Still on mute, Eric.
Eric, could you please?
So one has to ask, where's the money going to come from for GMP relative to GLP? And usually it's doing a good job in the preclinical, pre-IND phase that one can de-risk the points of getting enough cash to make a GMP prep. It's rather hard when you're still doing monkeys to have enough money for even 10 grams of a GMP preparation. It's a matter of, it's a chicken and egg operation when you're a startup as opposed to a large company. So that's the rationale for only making GMP when you're ready to go clinical.
Mhm.
Also, on the question of stability, I might say that we see good stability, but our most revealing assay is a really simple one. It's denaturing polyacrylamide gel electrophoresis, which we always find has better resolution for little impurities than an HPLC. And it's really easy to do it on our own bench as opposed to farming it out to a CDMO. I mean, this is something everybody ought to be aware of, particularly when you're starting to do stability studies.
Yeah. I just this last week I was in a conference and someone else threw the question of GLP material, and there was a heated discussion in the room with several people saying that there is not such thing as GLP material. GLP is just for analysis and experiments, not for production. I agree with you in several of the points. I mean, but also characterization. The standard for us, to be honest, is the LCMS methods. That's what gives you the most information regarding stability of your molecule, and then you're able to see, you're able to follow degradation in a controlled way. At the end of the day, these kind of therapies, they are going to be based on oligos that are quite well studied, and I think the challenge here is going to be to unify that information, as Sepe is doing in the analysis part, because if you design your program properly, you can justify a lot of things in a written manner. Now, the degradation pathway of oligos is known already. The things that you have to be looking for degradation is known. The stability of most oligos is quite long and not something that you have to worry. So it's about the combination, that knowledge that eventually will make the data package compiling enough for the authorities.
Pablo, I have just something else in my mind. I'm a biologist, and in my former life, I also developed oligonucleotides. And when I first approached the CDMO, they're asking me about acceptance criteria and what are your major impurities. And I really didn't understand even the words. So maybe for the biologists in the room, and I can say this, I'm also a biologist, what are the typical impurities for a phase one batch that you would ask for, or you would develop an analytical method for? Is it just N-1, N-2, or what are your considerations regarding impurities, just to explain it to the biologists?
Yeah, so that's the most straightforward decision to make. You do the N-1, N+1, and then if you have phosphorothioates, the PO impurity, so oxidation of one phosphorothioate to the phosphodiester. With that way, you ensure that the analytical methods is set to detect a peak and what immediately comes after and before. With the N+1 and N+-, and then the PO to make sure that you detect some of the degradations. And we know that the degradation of the product goes that way. I would say that for 95% of the oligo programs, that is enough, and you should not go much more than that. However, now we are seeing how the molecules, they are becoming more critical and more complicated. And when a company has platform data, they can leverage that and include some additional impurities. And that would be probably include something else that is going to be formed during the process that they know that is going to form with their molecules or so on. But for general projects, I would always say N+1, N-1, and PO impurity. In that way, you make sure that you get the best separation of So the best selectivity of the method, the differentiating changes of one base, and then you're able to detect degradation with the PO impurity.
What about something like depurination? These are co-eluting impurities. Hard to see. Would you also, in general, advise to identify them, or is it more like a specific for a specific compound?
Yeah, that's more for general compounds, and it really depends on which oligo you have and which chemistry you have for them to form. I've seen most programs, it doesn't really form the depurination impurity. It really depends on the conditions that you have in your process and what you expose the molecule to. And in most cases, it doesn't form by the process per se. But yeah, if it really depends on the chemistry that you're using and how you use it.
Okay, and another topic which borders or which did bother me in the past was biologists come always from high purity expectations in the oligo. What would you I'm a biologist, so what would you advise for a biologist as a phase 1 trial to keep or to have a straightforward CMC strategy which is sufficient for phase 1, at the same time allows to increase the quality for phase 2 and phase 3? What would you advise as acceptance criteria for an oligonucleotide?
Okay, I feel I'm really privileged to be able to say this to people. Every time that you see 90-something% purity with a generic method, it's a lie. Just understand that. A chromatographic method, you can, make it so bad that you only have one peak. That's usually not the truth. So yeah, I see how companies that they have never done any clinical development, they come with expectations of purity of like 99% purity. Like what? No, what are you saying? No, that doesn't work like that. For an early, I'm not gonna put a definitely number on them, but I've seen programs that were able to do a phase 1 with close, like below 80% purity. Why were they able to do that? Is because they had a tox batch that was even lower in purity. So they were able to justify that by tox studies that they could use the material. And then over development, the specification got tighter to over 80% and below than 90, and after for commercialization in phase 3, higher than 90% purity. So where you start, it depends on how complicated is your molecule, and that you are going to justify with the tox batch that you are going to use also.
Yeah.
And again, if you see 99% purity, it's a lie.
Yeah. So, and just one further idea in the room that what we see very often is you also need the potential to increase the quality in later phases because the authority expect higher purities in the phase 2 compared to phase 1, especially when you validate your process and you need more material for later on batches. It's also easier to upscale for the CDMO if the expectations start low and get better during the process.
Exactly. Yeah. Don't tie yourself with a very high specification at the beginning. There are consultants out there that can see your molecule and tell you exactly what to do. The higher you go at the beginning, the more complicated you're going to set the ground for you. So think about it carefully at the beginning. See what you can produce, see how complicated your molecule is, and on that, base a decision on what to use.
So, are there other questions in the space in the group that needs to be addressed to Pablo? Maybe last question from my side. When you get approached by scientists asking for a quote for phase 1, what would you propose as a typical timeline for a standard batch? I don't know, 100 gram, 50 gram, whatever. What is the typical timeline that you propose to the scientists?
Yeah. So the usual timeline, it's from the moment that I start working until the clinical trial, until I have the GMP material for the clinical trial, is 6 months. That means it's 3 months for internal developing the analytical methods, doing some process development, proof of concept in my manufacturing platform, and then the non-GMP and GMP. That depends more on the scientists that I have in front than on me. Yes, developing a method takes time. Validating the method takes time. That's what you need to release under GMP. Preparing a GMP batch only requires, as I mentioned, the method and a batch record, and then the manufacturing takes 2 days. So it's more because the client needs to review documentation, needs to understand what they are doing, and when you send them something to sign, instead of giving you in a week, they take 4. But yeah, it's most programs, when we start working with them until the GMP material is prepared, is 6 months.
Okay, very good. Should we ask Seppe? Do you wanna follow and step in?
Yes. So my part of the webinar today will focus on key outtakes we have written down in a CMC roadmap for N of 1 projects to guide investigators, patients, organizations, clinicians that will be applying a N of 1 treatment for the first time, to supply them with the information, minimal information they need to know to successfully and efficiently perform all the preparations. But, however, during the numerous lengthy discussions we had to make this roadmap, the answer was often, it depends. It's not a universal answer. So during my presentation, for a big part, instead of presenting you with answers, I will actually present you with questions. And if you can answer all the questions, then you are likely ready to start the clinical study with your patient. So I will first start off with the preclinical chemistry selection. If you're currently still, if you are starting on the development of an oligonucleotide or are in the early stages of the development, the first big guidance we can give you is, if it's possible, please stick with well-known and well-studied platforms. Not only do they typically offer a better potential for success and understanding of toxicity risks on a biological aspect, but a well-studied platform typically also means that there's more manufacturing experience on the market. And as Pablo earlier mentioned, a lot of GMP and manufacturing revolves around risk assessment. So if you have a compound which is a lot of manufacturing experience in, there is a severely reduced risk that anything unexpected happens and the timeline during the GMP process would extend and the patient would need to wait longer for the treatment. It also, another benefit is that it will typically also offer a wider selection of manufacturers. If you are chasing a novel compound, a novel modification of oligonucleotide that only one specific company has experience with, you will be at their goodwill. If they say they have no slot available for you this year, you will likely not be able to get your compound manufactured this year. This goes geographically as well. Typically, you want to move the compound around as less as possible, because every form of transport, just like taking your car to drive to the supermarket, has a risk of something going wrong. So if you can also minimize the geographical spread that the treatment needs to make, you minimize certain risks. And an example of such a platform approach was actually the original N of 1 oligonucleotide milasen. Timothy Yu used the blueprint of Spinraza, also known as nusinersen, to develop a splice switching oligonucleotide for Mila using the same chemical blueprint. And after the successful first treatment for Mila, multiple such splice switching oligonucleotides followed the same blueprint, like, for example, atipexor or ceftriaxone. Another suggestion, and this is in case your target has no existing platform yet, it is to consult manufacturers early on. They, based on their experience, they can often indicate if the design of your oligonucleotides will lead to problems during manufacturing. A classic example is repeating multiple guanines right after each other in one oligonucleotide. Another example can be the placement of conjugates. For example, if you want a GalNAc conjugated oligonucleotide to target the liver, biologically, it might not make much difference if it is at either end of the molecule, but for the manufacturing, this could make a big difference. And if you unfortunately chose in your early development to put it on the wrong end, this can lead to manufacturing issues to reach a certain quality later during clinical investigations. For the scaling strategy, one of the important messages is that the cost of manufacturing is not linear. So one of the largest costs for oligonucleotides is often the human resources. So it can be beneficial to inquire about scale ranges and limits and their respective prices. And it is often beneficial to maximize the capacity of a manufacturing train and combine the material you need for multiple studies in one manufacturing order. But as indicated earlier, due to financial constraints, that is not always possible, unfortunately. For toxicity studies, if you perform them with a contract research organization, it is also important to consider supply options. It might be possible that they can receive the drug substance as a bulk, the drug product as a bulk solution, or the drug product in individual dosage files. But the important thing here is to try to optimize aliquoting strategy so you do not have excess material left in each file after use, and you can minimize any wasted oligonucleotide. And another suggestion is to order excess material or even doubling the required amount when you are going to in vivo toxicity studies. This is to mitigate certain operational risks. For example, if there is material loss, there are spills, there are contaminations, or there are study repeats required or additional protocols or studies required, then you don't need to wait for the additional material and perform a second order, and you can immediately continue. But it is often also crucial to retain a dedicated portion of this batch for further reference so you can perform comparability testing between your toxicology batch and the batch that will be given to the patient. So you can prove that the impurities that are present have a similar profile. Another option here is to use the GMP produced batch that is intended for the patients and perform the toxicology studies in vivo with the exact batch that the patient will receive. So the route from the active ingredient to the patient, it always follows the same physical chronological workflow. You first complete our preclinical studies, then the drug substance is manufactured at a CDMO. After that, the drug product or the formulation is made at a hospital pharmacy or at a fill and finish CDMO. And finally, the final formulation is delivered to the patient. While this reflects the physical execution, it is not necessarily the best way to strategically plan the entire manufacturing. And in the next slides, I will propose a reverse engineered strategy so you can, at each step of the planning, provide as much information as possible so you can get as detailed feedback as possible so you can minimize the amount of time spent in meetings or waiting for emails. So the first step is assessing what are the patient demands, and the first thing is actually a strategic onboarding. At any step during the entire process and planning, consider partnering up with a consultant, whether it is to review anything or to assist when the matters get too complex. I know that nowadays, oftentimes, AI is used as a consultant, and in some cases it might help you initially, but very often it will also be misleading. So it is best to always have things reviewed by a human consultant. And the more relevant information you can provide to a consultant, the more efficiently they can help you. So the more information you supply to them, the more reward on investment you get from them. The next step is to determine what does the patient need for my initial clinical investigation. This can be defined as the dose times the dosing interval times the time span. For example, if over a time span of 5 years, I intend to dose the patient with 40 milligrams of oligonucleotide every 3 months, I will need approximately 30 vials of 40 milligrams of drug product. But this is the minimum amount of drug product that you need per patient. Then another important question is, what is the intended route of administration? For the manufacturing, this can severely impact some of the quality criteria that are set on the drug itself. And another question is, if you are ready to go into a clinical stage, are there any other patients that can benefit from this therapy? Because typically, as explained before, the costs do not increase linearly, so the cost per patient is typically less if you have more patients. The overall cost is more, but the cost per treated patient is less. Next up is the regulatory framework and formulation pathways. Next, once you know how you want to deliver the oligonucleotide to your patient, you can then define also what the regulatory framework is in which your clinical investigation is set. Examples could be a hospital exemption, compounding rules, or a standard clinical trial application or investigation on new drug. And one of the questions here is GMP-grade material actually required? In some situations, it is not required to have GMP-grade material. For example, in a name-based patient treatment in some countries in Europe. But if GMP-grade material is required, which clinical phase do we want to match or do we need to match? Because the specific requirements vary per clinical phase. And lastly, if no GMP-grade material is required, you still need to define what the minimal requirements for the intended route of administration in the given clinical setting are, so you guarantee patient safety. The next step, if you know the regulatory setting, you can evaluate the options for the preparation of the formulation. It might be possible to compound the drug at a hospital pharmacy, but most oligonucleotide treatments are delivered as injections with strict sterility requirements. So then the main question becomes, can the hospital pharmacy meet these sterility requirements, or what do they need to meet these requirements? And if they cannot perform this, it might be necessary to partner with a fill and finish CDMO that possesses the necessary infrastructure to guarantee the sterility of the drug product. After you've decided on one or multiple strategies for formulation, you can then contact the institution that's responsible for preparing the formulation, whether it's the hospital pharmacy or the fill and finish CDMO, and you can provide them with as detailed information as possible based on the previous steps. Examples are the type of treatment you will be formulating, what is the active ingredient, what is the route of administration, what is the regulatory framework of this treatment, what is the required amount of formulation. As previously mentioned, it is best quantified as I need X amount of vials with X amount of volume with X amount of active pharmaceutical ingredient in hand, meaning I need this amount after all required amount for quality control is subtracted. And if you are able to determine which tests and studies are necessary, this is also a good thing to share. The key questions you want answered from the manufacturer is, the first question is, what is your experience with this treatment or formulation? If they have no previous experience, it might be required that extra development studies are performed, and therefore it might be in financial and timeline perspective be more beneficial to partner up with another institute that has the experience that is required, as typically demonstrated experience is one of the regulatory minimal requirements. Another question is to inquire about protocols and tracking systems to guarantee chain of identity. This is important to make sure that no mix-ups occur and your patient is treated with the correct medicine. Examples of this are the master batch records, as Pablo mentioned earlier, but also etiquettes or labels that are put on the files, for example. The next question is, what additional studies, additionally to the studies you determined, do they deem necessary, or are there studies they deem unnecessary? And if some of the studies cannot be performed at the fill and finish themselves, it is often possible to outsource these studies to the manufacturer of the oligonucleotide. The next question is if they are able to perform an analytical method transfer. Sometimes analytical methods, as for example, for the determination of the purity, can be transferred between institutions and they do not need to develop an analytical method separately and qualify or validate these methods separately. And the next question, and this is one of the important questions, how much drug substance do you need to give me the required amount of drug product that I need at the end? They will have the best view of all material that is required for additional studies or stability studies, for example, and can give you a better overview of how much material they need in total to give you the amount of material you need. And then the last questions are, what is the expected timeline you can deliver me this? What are available time slots? And then you can ask as well for a quote or cost estimate for the manufacturing. In the next step, you can then take the information you have from the drug product manufacturer to the drug substance manufacturer. In this case, you can provide them with the same information, but you can also provide them with the exact amounts that the drug product manufacturer requires. As the required equipment and processes sometimes can vary between scales, so it is not always simple to quickly change the amounts at the last minute. And the key questions here are, again, to ask for the experience, to have demonstrated experience with manufacturing this kind of active ingredient or modality. Again, to inquire about the protocols and tracking systems that are in place to guarantee a chain of identity to prevent any mix-ups. Additionally, to the studies you determined, what additional studies do they deem necessary from a quality, safety, and regulatory perspective? And could specific studies that are required from the formulating side be outsourced to the drug substance manufacturer? Examples of these studies could be the extinction coefficient determination in formulation versus in pure water, or solubility studies. And then another question is, if the analytical methods that are developed at the drug substance manufacturer can be used to support as a method transfer or to support method development at the formulating institution to save time. And then again, the same two questions come. What is the required manufacturing time? What are the available time slots? And what would be a cost estimate for all the studies and the manufacturing of the material? And after you have all these questions answered, you should be able to create an overview and define a final strategy and scale. You should be able to establish an end-to-end chain of identity so you can guarantee the product traceability throughout the entire manufacturing. You can try to map the integrated manufacturing and study timelines to create a full timeline. But it is worth considering a slight excess of material to mitigate any manufacturing and handling risks. And an important thing is consider ensuring shipments. This is typically only 1% of the shipment price, but if something goes wrong, you at least are not at a big financial loss. And finally, it is always worth to have an expert review the full strategy and point out any holes that might still be present, or if you are ordering any excess that is not necessarily required. And if it's required, for example, with an IND submission, at this stage, you can submit your strategy to local authorities to obtain approval to start with clinical investigation. And once you have the approval, you can request a final quote and submit the order to start the manufacturing. One important message that I've tried to highlight is that there are more material demands beyond patient treatment. One of these examples is a stability study. These are mandatory to guarantee that the formulation retains the needed quality and remains safe for patient administration. And recently, I've seen a case example of a previous N of 1 clinical investigation where they had, with consultants, devised a minimal stability protocol, but this protocol still required a total of 100 files, 72 of which were actually required for the study protocol, and 28 were as backups. But there was only 30 or 40 files expected to be needed to treat the patient for 5 years, during which this stability study ran. So this is an example where you can see that it's possible that you need twice or 3 times the amount you need to treat the patient for guaranteeing that you safely treat the patient. So when initially determining the amount of material you need, the amount the patient needs is not the amount of drug substance that you need. But it is possible to inquire about the limiting factor for a stability study. For example, it is often a standard approach to take 100 files. So then you can ask if the limiting content for the required number of, if the limiting factor is the total amount of files needed for all the tests to have a statistic representative result, or if the content of the files is the limiting factor. If it is the physical number of files, it might be possible to split your drug product over 2 or more files to reduce the amount of product that is needed for a stability study, but you need to make sure that this does not significantly increase contamination risks, risking the treatment not being sterile when applied. And aside from these stability studies, there are also numerous analytical and manufacturing-related studies that are often required, but do not necessarily require GMP-grade material. So when you have decided on a final target, you could consider ordering a larger amount of material for the remaining non-clinical studies. And this excess material can then, in the following step, immediately serve various purposes, such as method validation batches, preliminary stability studies, or as a reference standard. And as a last piece, I've listed some historical minimum regulatory expectations. They might still be the case, but it is always possible that the requirements become stricter. An example is that the manufacturing process, no development or validation might be necessary, but it might suffice that the manufacturer provides a demonstrated understanding of the manufacturing process based on previous experience. There need to be control strategies in place for the drug substance and the final drug product to guarantee product quality and patient safety. This, one of the things in a control strategy, is typically establishing a list of minimum acceptance criteria for the drug substance, drug product, and any critical starting materials that are used to make the drug substance or the drug product. And typically, as Pablo also explained, a detailed list of critical process steps is detailed, and respective controls is listed to demonstrate that in these critical steps you make sure no mistakes are made. For an IND, it is also typically required that you have already preliminary stability data for the drug substance and the final formulation, which typically needs to cover at least 3 months. This can be done, for example, parallel to the toxicology studies. And analytical test methods for these studies initially often suffice when they have demonstrated feasibility and do not require further validation at this stage. And that was my current outtakes of the CMC roadmap.
Thank you so much, Sepa, and thank you so much, Pablo. I think that these talks were really wonderful, high level and practical explanations of the process overall, and we're so appreciative that you took the time to come today. Since we're well over time, I'd like to take this opportunity to encourage folks in the audience to come to Connections and please share your follow-up questions and comments. I'm sure Sepa and Pablo would be happy to follow up there. And also, if you can spare another couple minutes on our behalf, a survey will auto-populate when we close this meeting, and we would very much value your feedback. Thank you, everyone.